like to thank the organisers for the kind invitation to present some of our work today. So another long-term focus of our lab has been adoptive immunotherapy. Adoptive immunotherapy using tumour-infiltrating lymphocytes involves isolating these T cells from a large tumour mass, as shown at the top of this slide. It's not working. Yep. And then selecting and activating these T cells with growth factors such as interleukin-2, as shown here, expanding these tumour-specific T cells in large numbers and then reinfusing these T cells back into the preconditioned patient. So this approach has worked really well in melanoma, which is a very immunogenic tumour, and it's much easier to generate sufficient numbers of these T cells. For other cancers, it's not the case. So this sort of approach has remained ineffective for other cancers. So work in our lab and others have used a genetic modification approach involving expression of chimeric antigen receptors, or CARs as we designate them, into T cells, as shown here, which can specifically recognise tumour-associated antigens. So the CAR consists of these extracellular single-chain antibody, consisting of the variable heavy and light chains of the antibody linked together. And this is joined onto T cell receptor signalling domains, which can very effectively activate the T cell. A lot of work in our lab has shown that T cell receptor signalling domains, including the CD28 co-stimulatory domain linked to the T cell receptor zeta domain, can very effectively activate the T cell. Other groups have shown other domains, such as 4-1BB, are quite as effective. So in the last 5 or 10 years, there's been a surge of clinical activity using these gene-modified T cells, or CAR T cells, in both blood cancers and solid cancers. There's really been some tremendous results achieved in some blood cancers, such as ALL and CLL, targeting antigens such as CD19. Some biological responses or anti-tumour activity has been shown in some solid cancers, but not to the same degree as these blood cancers. And it's really a challenge for the field to be able to extrapolate the results seen in these blood cancers in solid cancers. So really the promise of this type of approach is really highlighted in this slide here. This little girl here, Emily Whitehead, who was diagnosed with ALL as a 5-year-old in 2010. And she underwent chemotherapy and bone marrow transplantation, but unfortunately relapsed. The disease relapsed in this patient, and she was enrolled into a CAR T cell trial targeting CD19 antigen in 2012, and I can gladly report that she went into remission, and she still remains free of disease today. So up to 90% response rates are now being achieved in these type of patients. So I think the work of Carl June and others have really pioneered this type of approach, and it really does highlight the tremendous promise of CAR T cell therapy as an effective cancer treatment. So today I'm going to talk about a recent phase 1 clinical trial we just completed at the Peter MacCallum Cancer Centre. So we look to expand this approach against other antigens. And this trial involved targeting CAR T cells against the Lewis-Y carbohydrate antigen in acute myeloid leukemia patients. Now, we haven't tested this in breast cancer patients, but we are keen to test this next year in solid cancers, given that Lewis-Y is very highly expressed in epithelial cancers such as breast, prostate and lung cancer. So AML is a devastating disease. It's have a 5-year survival of less than 10%, and there's 12,000 new cases diagnosed in the USA alone each year. So the Lewis-Y antigen that we're targeting is a glycosylated carbohydrate. It's found on a variety of proteins and lipids. We believe it's a great target for immunotherapy because it's overexpressed on approximately 60% of carcinomas, about 10% of AML and myelomas. Its expression is associated with poor prognosis. It is expressed at varying degrees on some normal cells or normal tissues such as the intestine and on neutrophils. So it's very important to perform a phase 1 clinical study to test the safety of this approach. We were fortunate to obtain the humanised antibody from Andrew Scott's team, a single chain from Ludwig, and we proceeded to construct the CAR using that humanised single chain. So we're linked on the CD28 and T-cell receptor zeta domains onto this single chain. So as preclinical development of this approach, we were able to show in non-skid mice that adoptive transfer of human T-cells engineered with this CAR targeting Lewis-Y could very effectively inhibit the subcutaneous growth of the ovarian carcinoma cell line, OFCAR3, as shown here. Just died on me. This is compared to transfer of control T-cells that had little effect on the growth of these tumours. So in this experiment, human T-cells were transferred on day 7, 8, 9 and 12 against established cancer. Cheers. Okay, so we went forward to perform our phase 1 clinical trial here. The primary objective was to test the safety, of course, of this approach, and as secondary objectives we looked at persistence of our gene-modified CAR T-cells and the trafficking to the site of disease. We measured serum cytokines and also measured whether there's any anti-cancer activity from these CAR T-cells. So 5 patients were treated in total. They received from 5 × 10⁸ to 1.3 × 10⁹ T-cells, and each of these patients were preconditioned with fludarabine to allow engraftment of these transferred T-cells. 10% of the cells were also labelled with the tracer indium to better monitor persistence and trafficking of these T-cells in the patient. So patients were pre-selected on Lewis-Y expression on the AML blast cells, and the patients had to have at least 25% expression of Lewis-Y in these blast cells. The patients ideally had to were in cytogenetic remission after chemotherapy. So after enrolment, we isolated the PBMCs by apheresis. The CAR anti-Lewis-Y single chain was inserted into these T-cells by retroviral transduction. So these cells are activated to allow efficient transduction. These cells were then expanded ex vivo, as shown here in these large bags here, tissue culture bags, and then reinfused back into the patient. So this slide here just shows the summary of the results we saw in these 5 patients. So patients 1, 2 and 4 were under cytogenetic remission after chemotherapy. Patient 3 here had active leukemia at the time of transfer, so there's still 70% blasts in the bone marrow here. Patient 5 had 11% blasts in the bone marrow. As I just mentioned, patients received between 5 × 10⁸ and up to 1.3 × 10⁹ T-cells. And the first point to make is that we saw no adverse event in any of these patients, so the CAR T-cell trial was safe. Patient 2 experienced slight neutropenia, grade 1 and 2, and this resolved without intervention. And there was some skin rash and a slight fever in patient 3. So the best results we found were in patient 2. So this patient was under cytogenetic remission for up to 22 months after transfer. And this correlated with long-term persistence of these CAR T-cells in the patient. I'll show you that data shortly. And even patient 3 here, who had active leukemia at the time of transfer, we saw a biological response in these patients where we saw a transient decrease in circulating blasts after CAR T-cell transfer, which is quite encouraging. So by RT-PCR, we're able to show that these CAR T-cells could persist in the patient to varying degrees. And where I just mentioned patient 2 that showed the best biological response, we saw that these CAR T-cells persist up to 10 months in the bone marrow and the blood. So you can see here, after 10 months, you can still see the presence of the Lewis-Y transgene in the bone marrow and blood of these patients. This patient. Correlating with anti-cancer activity seen in these patients was we saw a spike in cytokine levels in the serum of all patients. This is representative data from one of these patients. So we saw a transient spike in important cytokines such as interferon gamma, IL-2 and IL-4 in all the patients. And as I mentioned earlier, we used some of these cells, about 10% were labelled with indium tracer. We're able to monitor the trafficking of these CAR T cells after transfer. And this is representative data from one of these patients, patient 2. And you can see clearly here that shortly after transfer, so it's about 30 minutes after transfer, we can detect the presence of these CAR T cells in the lung. And around about 4 hours after transfer, you can start to see these T cells migrating to the spleen and evidence of migration into the bone marrow here, which is quite evident after 15 hours here. We saw similar results in all patients treated. What was a very interesting observation was that one of the patients developed leukemia cutis shortly after, had a prior history of developing leukemia cutis and developed a skin rash shortly after transfer of these CAR T cells, as shown here. So we proceeded to biopsy that site, and were able to detect the presence of AML blast cells, as shown here, indicated by the CD33 positive staining. And also, in that site, we could detect the presence of CD3 positive T cells at that site. On further analysis by RT-PCR, we could detect the presence of the Lewis Y transgene within those T cells at the site. So not only did our CAR T cells had the capacity to migrate to the bone marrow, they could also migrate to other sites of disease, including the skin. So it was very encouraging results. So today, in this short presentation, I've shown you that we were able to very efficiently generate human T cells expressing the CAR, targeting the Lewis Y antigen, and these cells could mediate anti-tumour activity in the mouse model. I've shown that in a phase 1 trial that the transfer of these CAR T cells was well tolerated in all patients treated. We saw biological responses in all patients following CAR T cell therapy. And importantly, these CAR T cells persist for a period of weeks and traffic to disease sites. And we're now gearing up to perform a second trial starting 2016, targeting patients with solid cancers, including lung, breast and prostate, where Lewis Y is very highly expressed. So we're very excited about starting this new trial. Only about 10% of patients express that Lewis Y, AML patients express Lewis Y, so we're very keen to start this new trial. And we've also found in our mouse models that combining checkpoint blockade with CAR T cells is very effective in inducing reduction in tumour growth. And so we're very keen in even further trials to combine CAR T cells with immunotherapy such as anti-PD-1. So it just leaves me to thank the people who were involved in this work. I'd just like to thank the members of my laboratory and Mike Kershaw's lab, and, also, and very importantly the clinical people who were involved in this trial, Miles Prince, David Ritchie and Paul Neeson, that enabled this trial to be undertaken. I'll just leave it there. Thank you.
And we'll take a couple of quick questions for Dr. Beavis and Dr. Darcy now. I have one. I have a couple actually. So one question I have is, you know, I always feel like the CAR T cell therapy has a dirty little secret, which is that it works very well for liquid tumours, but it's been pretty ineffectual up to this point with solid tumours. So what do you see the future of this therapy in terms of moving it into solid tumours, like breast cancer, for example?
Yeah, well, I believe, so we're sort of tackling other issues in terms of access to these CAR T cells to solid cancer sites, and then also the immunosuppression networks are set up in these various cancers. So I think we're starting to now, as Paul just showed you in his talk, understand much better these immunosuppression networks that exist, and I think we have now reagents that can actually reverse that. And so I'm very confident that in future combination trials that targeting pathways such as PD-1 or CTLA-4 combined with CAR T cell will end up being very effective.
So you feel that even though apparently they're not terribly effective at penetrating solid tumours, at least you can make certain the ones that get there are working, right? By using checkpoint inhibitors, remodelling the tumour microenvironment.
I think it's important to note that there has been some responses targeting some antigens such as GD2, for instance, using CAR T cells. It's not as, the results in some of these blood cancers have been incredible and really put this therapy at the forefront. But I believe that we are seeing some biological responses, and as a lot of room for improvement with these combinations.
I think the other thing I'd add to that is there's some approaches now preclinically where they're engineering their CAR T cells to express certain chemokines within the tumour microenvironment, so you can really amplify up that system. And they do it in a way where it's tagged to like an NFAT promoter, so that when the CAR T cells get activated, they only make the cytokines at the site of the tumour antigen. So I think those kind of approaches will be useful for solid tumours.
Okay, we'll move on. I think Dr. Eichmuller.